We're going into the world of cellular energy.
Oh, yes.
Fascinating stuff.
It is.
Yeah.
We're talking glycolysis.
Glycolysis, the breakdown of glucose.
How our bodies turn fuel
It's amazing.
into usable energy.
Yeah.
And
It's more interesting than you might think.
way cooler
Oh, absolutely.
than I thought it would be when I started looking into this.
Yeah.
We're going to break down glycolysis
Step by
step by step.
10 steps.
10 steps.
Yeah.
Lots of enzymes.
Enzymes, phosphates,
Phosphates.
all sorts of molecular magic.
Molecular magic.
It really is.
It's going to be great.
Yeah, you know what's really cool about glycolysis is that it's ancient.
Yeah.
I mean, this process...
What ancient are we talking?
I mean, we're talking about a process that's been around for billions of years.
Billions.
Nearly every living organism on Earth uses it.
So
It's like a universal language.
bacteria are doing this.
Oh, yeah.
Plants are doing this. Plants, animals, fungi,
Everything.
doing the same thing.
Yeah, it's fundamental
Wow.
to life as we know it.
That's wild. So this is like
You think about
a
it.
really important thing to understand then.
It is. It's the basis of how we get energy from the food we eat.
And it's actually kind of surprising to me that it's the same in all these different organisms.
I know.
That's really interesting.
You would think there might be variations, but the core process is remarkably conserved.
Yeah. Well, and I was reading
Yeah,
that glycolysis actually uses energy
it does.
before it makes more energy.
That's right. That's one of the fascinating things about it.
I don't get that.
Well, it's like an investment.
Okay.
You have to put in a little energy up front
Yeah.
to get a much larger payoff later.
So like priming the pump.
Exactly. Priming the pump.
But before we get into that,
Yeah.
I think we need to zoom out and get like a big picture
Yeah, let's set the stage.
of what glycolysis is.
All right, so glycolysis,
It's essentially a 10-step biochemical pathway
10 steps.
that takes glucose,
Okay.
which is a simple sugar.
Glucose, everyone knows glucose.
Right. It's our primary source of energy.
Blood sugar.
Blood sugar, exactly.
All right, so we start with glucose.
Start with glucose and we break it down
We break it down.
systematically to extract energy.
Okay, where does this all happen?
This all takes place in the cytoplasm,
The cytoplasm.
which is that busy space outside the nucleus of a cell.
Okay,
It's like the cell's main workshop.
gotcha. So not in any special little compartment.
Nope, right in the cytoplasm.
Right in the cytoplasm, the
Well, that's
workshop.
where the action happens.
All right, well, let's get into the nitty gritty.
Let's dive into those 10 steps.
Step 1, what is it?
Step 1 is phosphorylation.
Phosphorylation. And what's going on there?
So imagine you have a heavy box.
Okay.
You're trying to move it.
Yeah.
Adding handles makes it easier, right?
Oh, yeah.
That's kind of what phosphorylation does to glucose.
Okay.
We're adding a phosphate group to it.
A phosphate group, what is that?
It's a molecule with a phosphorus atom
Okay.
and some oxygen atoms attached.
All right.
And the energy to do this
Yeah.
comes from ATP,
ATP.
our cellular energy current.
Oh, yeah. Adenosine triphosphate.
That's it.
Right.
Exactly.
So we're using ATP to add
to add a phosphate
a phosphate to the glucose
to glucose to make it
more reactive.
more reactive. Okay, so it's like we're giving the glucose molecule a handle,
You got it.
so it's easier to work with.
Precisely. That phosphate group,
Yay!
it destabilizes the molecule.
Destabilizes.
It's like winding up a spring
Oh, okay.
to unleash its potential energy later.
I like that analogy.
Yeah, it's a good way to visualize it.
So we've got this enzyme. What was it called again?
Hexokinase.
Hexokinase. It's using ATP.
Yes.
It's adding this phosphate
to the glucose.
to the glucose, and we're making the glucose
More reactive.
more reactive.
Ready for the next step. All right. On to step 2.
Step 2,
What do we got?
isomerization.
Isomerization.
Now, this one might sound a little complicated,
Oh, yeah.
but it's really just a rearrangement
Oh,
of
no.
the atoms within the glucose molecule.
Okay.
We're turning glucose
Yeah.
into fructose 6-phosphate.
Fructose.
It's another sugar.
Is it like high fructose corn syrup?
Not exactly. It's a different kind of fructose molecule,
Okay.
but it's still a simple sugar.
And why do we need to do that?
Well, it's all about preparation
Okay.
for the big split that's coming up.
Split.
Yeah, remember we're aiming to break that 6-carbon glucose
Yeah.
into 3-carbon molecules.
Gotcha.
So this isomerization,
Yeah.
it's just prepping the molecule.
Keep lining things up.
Exactly,
Right.
to make the split easier.
And what's the enzyme for this step?
This one's called phosphoglucose isomerase.
Phosphoglucose isomerase.
Big word.
Big word. But it's just rearranging the atoms.
That's right. From glucose to fructose 6-phosphate.
Okay. Step 3.
Step 3, back to phosphorylation.
Phosphorylation again. Okay.
We're adding another phosphate group.
Another one.
This
back.
time to that fructose 6 phosphate.
Yeah.
And the enzyme responsible for this
Yeah.
is phosphofructokinase.
Phosphofructokinase.
Another mouthful,
Another mouthful.
but it's a key regulatory enzyme.
Okay.
It controls the flow of molecules through glycolysis.
So we're using another ATP here,
Yes,
and we're adding another phosphate.
correct.
Okay, so now we've got fructose 136 bisphosphate.
That's it.
What's bisphosphate mean?
It means there are 2 phosphate groups attached,
Oh, okay.
one on each end of the molecule.
So we've loaded this thing up
We've loaded it up
with phosphates.
like the Roundup spray.
Yeah, ready to unleash
We've invested energy.
2 ATPs in.
Exactly.
And now we're ready to break it apart.
You got it.
Step 4.
Step 4.
What happened?
Things start to break apart.
Here we go.
This is where aldolase.
Aldolase.
The enzyme aldolase comes in
Okay.
and cleaves that 6-carbon sugar.
Finally, we're splitting it.
We're splitting it
Right down the middle
right down the middle.
into 2.
2 3-carbon molecules.
Okay, what are those?
We get dihydroxyacetone phosphate,
Dihydroxyacetone phosphate.
DHAP for short,
DHAP, okay.
and glyceraldehyde 3-phosphate,
Tetraldehyde 3-phosphate.
or GA3P. GA3P.
So 1 becomes 2.
We've got our 3-carbon molecules.
And this is a major turning point
Okay.
because that initial investment of energy
The 2 ATPs.
is about to start paying
All
off.
right, I'm excited.
Get ready for the energy payoff phase.
Let's go to step 5.
Step 5.
What happens?
Well, we've got these 2 molecules, DHAP and GA3P,
Yeah,
but the cell really wants to work with just one type.
to simplify things.
Yeah, for efficiency.
See how it happens.
So DHAP
Yeah.
gets converted into GA3P.
So we're doubling up on GA3P.
Exactly.
Okay.
And the enzyme that does this is called triosphosphate isomerase. Triosphosphate isomerase.
Another long one.
They're not making these easy to remember.
But don't worry about memorizing the names.
Yeah. Okay.
It's the concept that matters.
So now we've got 2 identical GA3P molecules
2 of them ready to roll.
ready to move into
The energy payoff phase.
the energy payoff phase.
Where things get really interesting.
All right. I can't wait.
Let's take a break and we'll pick it up there.
Sounds good.
Stay with us. Okay, so we're back and we're ready for the payoff phase.
I'm ready.
Where the cell finally gets some of that energy back
Yeah.
and that initial investment.
I'm ready for some return on that investment. 2 ATPs worth.
Exactly. 2 ATPs we put in, and those 2 GA3P molecules are prepped and ready to go.
All right. Hit me with step 6.
Step 6, we're talking oxidation.
Oxidation. Okay. Now I remember from chemistry
Yeah,
that oxidation is loss of electrons.
you got it.
But what does that mean?
Right, in this context.
Yeah, this context.
We're going to be stripping those high energy electrons from GA3P,
Okay.
So-
Stripping them away.
capturing sparks
Stealing energy.
from a fire.
Okay.
The enzyme that does this is glyceraldehyde 3-phosphate dehydrogenase.
Glyceraldehyde 3-phosphate dehydrogenase.
Long name, I know,
Wow.
but it's a key player.
Okay, and what happens to those electrons?
They don't just disappear.
Yeah.
They get picked up by a molecule
Okay.
called NAD+,
NAD+.
which then becomes NADH.
NADH are another molecule to keep track of.
Yes, but think of NADH like a tiny battery.
Okay.
It's storing those electrons
Okay.
for later use.
For later. Got it.
We'll see where that NADH goes later on.
Down the line.
Exactly.
Okay, so we've oxidized GA3P.
We have.
We've created NADH.
Yes.
And now we're on to step 7.
Step 7, this is where we finally get some ATP,
Finally.
the payoff,
Payday.
through a process called substrate level phosphorylation.
Substrate level phosphorylation. That sounds complicated.
It's not as bad as it sounds.
Okay.
Basically, a phosphate group is transferred directly from the molecule 1-bar-of-3-bisphosphoglycerate to ADP to form ATP.
Okay, so it's like a direct energy transfer.
It is.
Boom, ATP.
Boom.
We made it.
That's right.
So the cell's getting some return
It
in its
is.
investment.
Finally, it's starting to make back.
Okay, what about step 8?
Step 8 and 9
8 and 9.
are kind of a setup
Okay.
for the final payoff.
Okay.
We're going to move that phosphate around,
Okay.
remove a water molecule,
Remove some water.
and create a molecule called PEP,
QTV.
phosphenylpyruvate.
Phosphenylpyruvate. I'm not even going to try and remember that one.
You don't have to,
Okay, good.
but it's important because we've supercharged that phosphate bond.
What do you mean supercharged?
We've stored up even more energy in that bond.
So steps 8 and 9
Yeah.
are all about
Setting up
setting up
for the final,
for the big energy release.
the grand finale,
Step 10.
step 10.
This is where it all comes together.
Okay, lay it on me. We've got our supercharged PE and an enzyme called pyruvate kinase.
Pyruvate kinase, that's the enzyme for this CEP.
That's right.
Yeah.
And pyruvate kinase facilitates the transfer of that phosphate
Okay.
from PAP to ADP
To make ATP?
to make another ATP.
Another ATP.
Yes.
So we've got
We've got 2 ATPs now.
2 ATPs from each of those 3 carbon molecules,
Exactly.
plus those 2 NADH
You got it.
we made earlier.
Don't forget about those.
That's pretty good.
Yeah, it's a decent payoff.
So we started with 1 glucose molecule.
We did.
We invested 2 ATP.
Yes.
And now we've got a net gain of 2 ATP,
Correct.
2 NADH,
Plus 2 pyruvate molecules.
2 pyruvate molecules. What are those?
Pyruvate is a key molecule.
Yeah.
It's kind of at a crossroads
Okay.
of many metabolic pathways.
So it can go in different directions.
Exactly, depending on what the cell needs.
What do you mean?
Well, if there's plenty of oxygen around,
Oh.
pyruvate can be shuttled into the mitochondria.
Mitochondria, the powerhouses of the cell.
That's right.
Yeah.
And enter the citric acid cycle,
Citric acid cycle. What's that?
another energy producing pathway.
Oh, so there's more.
There's more to come.
We're not done with energy production yet.
Not even close.
All right. I'm intrigued. Let's dive into that next.
Sounds good. Let's talk about what happens to pyruvate when oxygen is plentiful.
All right. So pyruvate can go into the citric acid cycle.
Yeah, if there's enough oxygen around.
Okay. So this is like what happens when I'm just like, sitting here talking to you
Exactly, when your cells are working aerobically
aerobically with oxygen.
with oxygen.
So the pyruvate goes into the mitochondria
Right, the mitochondria are like the powerhouses of the cell,
and it enters the citric acid cycle.
also known as the Krebs cycle.
Krebs cycle, citric acid cycle.
Okay. But essentially what's happening is we're going to be stripping away more electrons
More electrons.
from the pyruvate,
Okay.
and those electrons are going to be shuttled
Where do they go?
to the electron transport chain,
The electron transport chain.
which is where things get really interesting because that's where the bulk of the ATP is produced.
So this is like the main energy production.
This is the big payoff.
This is where we're really making the big bucks.
Exactly.
And wait, is this where the NADH comes in?
Yes.
The stuff we made earlier?
Remember those little batteries we talked about?
Yeah.
The NADH molecules?
Yeah.
They're going to deliver those high energy electrons
Oh.
to the electron transport chain.
So they're like little shuttles
Exactly. They're carrying those precious electrons.
to the electron transport chain. And then what?
Well, the electron transport chain uses those electrons
Okay.
to create a proton gradient.
A proton gradient. What's that?
It's basically a difference in concentration of protons,
Protons, those are like,
hydrogen ions,
hydrogen ions. Okay.
positively charged particles.
Okay.
And this gradient, it's like building up potential energy.
like water behind a dam.
Gotcha.
And that potential energy is used to drive the production of ATP.
Oh, so that's how we get the ATP.
That's the magic.
Wow, so it's all connected.
It is. All these processes work together.
Yeah, amazing. So in the presence of oxygen,
Yeah.
pyruvate goes to the mitochondria.
It goes.
It enters the citric acid cycle.
Yep.
More electrons are stripped away.
Right.
NADH shuttles those electrons
The electron transport chain,
to the electron transport chain.
where a proton gradient is created.
Yeah.
And that gradient drives ATP production.
ATP, the energy currency of the cell.
Exactly.
Amazing. But what happens if there's not enough oxygen?
Good question.
Like when I'm exercising really hard?
Yeah, when your muscles are working anaerobically
Anaerobically without oxygen.
without enough oxygen to keep up with the demand.
Right. And that's when I feel that burning sensation.
That's lactic acid.
Lactic acid, I've heard of that.
It's a byproduct of fermentation.
Fermentation.
Right.
Like making beer. Kind of, but in this case,
Okay.
it's happening in your muscles
Okay.
when there's not enough oxygen.
So pyruvate can't go to the mitochondria,
No, it gets diverted
and it gets converted to lactic acid.
right there in the cytoplasm.
Okay, and that's what causes the burning.
That's the burn you feel.
And fermentation was the point of that,
Well, it's a way for the cell to keep generating some ATP.
even without oxygen.
Exactly. It's not as efficient.
But it's like a backup system.
Exactly. A backup energy pathway.
Wow. Our cells are so clever.
They are. They've evolved these amazing ways to survive.
So we've got glycolysis.
Yeah.
Then depending on whether there's oxygen,
Yeah, 2 different paths.
we go to the citric acid cycle and electron transport chain,
If there's oxygen,
or we go to fermentation.
if there's not enough oxygen.
And it's all about producing ATP.
The fuel that powers our cells.
This has been incredible.
I know, it's mind-blowing to think about all this happening inside us right now.
Right now, in every cell,
Billions and billions of times per second.
it's just amazing. Well, I think that's a great place to wrap up this deep dive.
I agree. We've covered a lot of ground today.
We've gone from glucose to pyruvate
Yeah.
to ATP,
We've explored the different pathways,
aerobic and anaerobic,
and we've seen how it all connects.
to keep our bodies running.
Exactly.
Well, thank you for joining us on this deep dive into glycolysis.
It's been a pleasure.
It's been an absolute pleasure, and I'm sure we'll be back to explore more of the fascinating world of cellular energy in the future.
Absolutely. There's always more to learn.
Until next time, keep those metabolic fires burning.